Welcome viewers to Gateway Learning Center YouTube channel. Today we're looking at biology under the title genetics. So under genetics, we'll only look at hemophilia. So we'll talk about hemophilia, and apart from hemophilia, we'll also look at blood group. Okay, so those are the two focusing area under genetics, which we'll discuss on today. So we understand that hemophilia, the first thing is it is a sex-linked disease, meaning that if it is a sex-linked disease, this hemophilia is inherited. It is inherited from the parents. Then the same hemophilia, we need to understand that hemophilia is a genetic blood disorder. So since it is genetic, then it is a blood disorder. Hemophilia is a genetic blood disorder where blood does not clot easily. Now, for blood not to close or to clot easily, we need to understand that there are some factors that causes this disorder. So one of the factors that causes blood not to clot easily. So some of the factors, we know that factors that causes hemophilia. One, we'll talk about fibrinogen. Fibrinogen is one of the disorder. Then apart from fibrinogen, we also have what we call prothrombin. Prothrombin is also another factor that causes blood not to clot easily. Then apart from this one, we also have what we call ionized ions. Now, when you talk about ions, you're looking at calcium. You're looking at calcium ion, which is also a factor. Then we can also look at another one. Of course, there are many. We can talk about tissue thromboplastin. So tissue thromboplastin is also a factor that causes this disease called hemophilia, where blood is not clotting easily. Okay, now apart from the factors and the definition, since we have known that hemophilia is a sex-linked disease, we'll talk about the alleles which are normally used whenever doing the genetic diagrams under hemophilia. So under the alleles, we'll start with a male who is normal. So normal male as this allele. So put capital letter H representing normal. Then of course male. Now at this point, this male will become hemophilic. So if someone is hemophilic, then the alleles will have X, then small letter H and Y. We know that the sex of a male is determined by Y. Then we'll talk about the female. So a female who is normal will have these alleles. Okay, so the female who is normal will have X, then H, then again X, H. So this female is normal. So female, normal. Then female who is hemophilic will have this part, X small letter H. Let me use the other marker. So X will have X small letter H and X small letter H. This female is hemophilic. Then again, female who is now at this point a carrier. A carrier now will have X with capital letter H and X with small letter H. So these are alleles which we use whenever we are doing with the genetic diagrams under hemophilia. So the other question which likely to come at the exam is why is that hemophilic is common to males than female. So when look at the alleles which we have here, this Y has no gene. Therefore, it is only this X chromosome which is containing this gene. So if this X is hemophilic, it has no other chromosome which contains another allele or gene for hemophilic which can protect it. So if one X is hemophilic, the other allele or the other gene will have no chance to protect the other one or to support the other one. Therefore, a male will be exposed to hemophilic easily than female. Now, in females, since X is the one which is responsible in carrying this gene, if this one, like a carrier one, if this X is hemophilic, then it has this other one which is normal, which will protect this one. Hence, a female won't be easily exposed to this disease called hemophilia. So this is the reason why males are more likely to suffer from hemophilia than female, because X has only one chromosome. When that chromosome is affected, then it won't have another one to support it. While females have two X chromosomes. So if one chromosome is affected, then the other one will support it. So that is the reason why males are more exposed to hemophilia compared to males. Now we look at the genetic diagrams. So I'm going to answer questions from the past papers using the knowledge which you have now. Okay, so this is the question from 2023, paper 2 GCE. A hemophilic mother had two sons who were both hemophilic. The father was non-hemophilic. So this means that the father was normal, while the mother was not a carrier, but she had hemophilia. So define hemophilia. So we said hemophilia is a condition or is a disease or is a bleeding condition which is inherited, where normal bleeding is not taking place due to lack of clotting factors. So this one had 1 mark. That's about hemophilia. So this was very simple. Now B, using the letters X, with a superscript small letter h and X with a superscript small letter capital letter H. Write down the genotypes of the father and the mother. So let me create space here by removing this part so that I can use it to explain. Now we're looking at the term genotype. Genotype is the genetic makeup of a living organism which determines how that organism will be. So the genotype of the father, since we have only been given these symbols to use, but we cannot only use one symbol. So if you can go back from where we started from, I was saying that whenever we are dealing with this part, you will discover that when someone is hemophilic, so in this case the mother was hemophilic, so female, they have 2x. Then since she is hemophilic, you put a small letter and a small letter. So this was for the mother. So these are the genotypes for the mother. Then as for the father, since we know that X and Y is for male, the father was normal, so we're going to put one capital letter H. So these were the genotype for the father and the mother. So we are done with that part. Now we can continue to look at another question. So using a genetic diagram, explain why both boys were hemophilic. So we're going to use these similar or same genotypes we have found to answer question C. So this information now is outdated. So we'll remove it. Okay. So a genetic diagram. We know the symbols. We have the symbols already, which are here. So we'll say first parental phenotype. So phenotype is the outward expression. So we know that we have the mother, then close with the father. So that is what we have as the parental phenotype. Then first parental, so I'll just use my dots. So first parental. Now in this case, we're going to put genotype, the genetic makeup. So we have already the genotypes. So where there is mother, we'll put these for the mother. So don't forget to put this cross, which means the reproduction is taking place there. Then for the father, so that is how we come up with a genetic diagram. We are building it up in a standard way. Okay. Then from here, we'll put the gametes, the sex cells. If you want between the first parental genotype, you can put random fertilization, where you don't know what will happen after the fertilization has taken place. Then from here, we're going to split these genotypes to form the gametes. So we have this, and I'm going to circle it. another one. We're going to circle it. I think I'll run out of space here. So I want to draw a neat one. So let me reduce my handwriting. So gametes will be here. Hope you're able to see. Okay, gametes. Then I'll get my genotypes. The one will be here. So this is for the mother. Then you circle them nicely like this. Let me use another color for the father, so that when you're plotting, it will be very easy for us to see. Then from here, we're going to get for the father. At this point now, don't put this cross. So for the father, phone is busy. So for this one is for the father. Then I'll circle it. Then of course Y, I'll also circle it. So those are my gametes. Then from here, we'll put the offspring. So first offspring. Okay. So first offspring. Okay. So what will happen now is this gamete will give us 2 parts, and these gametes will also give us 2 parts. So we know that. Okay. We have X with a small letter, then also have X with a small letter H. Same here. We have X with small letter H, like that. So the 2 has given us 4 parts before these 2 comes in. That is a simple way I've learned it. After this now, we'll do a simple part, which is you drop this part, which is Y, like that. So after you drop this one, you jump the second, you go on the third. So drop, jump, drop. Therefore, the other one will come from here and to go all the way up to here. And it's also produce Y. So drop, you jump, then you drop. Of course, this one which has remained will fill up where the missing alleles are. So from here, from there, now, okay. So from there, now this part will drop here. Then you have something like this. Then this one also drop there. Then you have something like that. So that is how we plot the genetic diagram. So this is, we have a boy, here we have a girl, here we have a boy, here we have a girl. So despite my space was small, so they're looking like they are too close to each other, but hope you have understood. So you drop, jump, you drop. Same applies here. When you drop here, you're going to jump and then you drop on the other part. So this is the phenotypic uh, phenotype which we're talking about. So we have shown that this boy was hemophilic and this boy was hemophilic. So the ratio for boys who was hemophilic were hemophilic and the girls were, so we have a carrier and a carrier. So it is 2 to 2, which is equal to 1 to 1. So this is how we draw a genetic diagram under hemophilia. Next, we move to another question on the same part of hemophilia.